So basically you have all of the main building blocks now. We've talked about search strategies, we've talked about energy functions, low res, high res, prediction and design. So you have all these components. After that, it's kind of combining things together into some larger applications and making certain functions and predictions happen. So I think that what you're going to see is a combination of these different tools into different problems from here on out, and then we'll switch a little bit to thinking about what problems do you want to solve and pick some projects for you to do so you have these pieces to pull together. So the next thing I wanted to talk about was my favorite problem in the field, which is protein-protein docking, taking two proteins and figuring out how they fit together. Well, actually, docking in general would be taking two molecules and pulling them together, and there's a few different subclasses. There's small molecule docking, where you're taking something like aspirin or a hormone and docking that into a protein. And what's important there is the small molecule has a lot of flexibility, and modeling that molecule might be a little different than proteins. There's also peptide protein docking, where you have a short strand of protein, maybe 8 or 10 amino acids, and there the backbone is going to be very floppy, and so moving the backbone is going to be pretty important as you dock it to a protein. And then there's complete full protein docking. And what's nice about that is if both proteins have folds, then that structure, the backbone structure of the protein is often pretty stable, and so you might not have to move the backbone as far, and you're mostly looking at side chain flexibility. So that's why protein-protein docking is one I like to do, because it restricts some of the problems you have to deal with. Yeah, so we've talked about a bunch of these problems. So if you had to throw a protein docking algorithm together, what would you do? Could you think about how you might pull that together? All right, this is good. You guys have a lot of ideas. What do you guys have?
So I guess we kind of said if you have two proteins that you assume they're going to dock together, you haven't making any assumptions, so we assume that maybe the backbone's not changing. Okay. So that makes things a lot easier.
Yes.
And then you have to guess where the surface on each protein interact. Yeah. So then you calculate the delta G for the two proteins by themselves, and then you combine them, and then you calculate the energy that you would gain by basically having these surfaces interacting. Okay. And then you would use the rotamer library to try to make the residue, have the maximum gain energy as you combine. So then kind of that maximum decrease in energy kind of be your docking situation.
Yep, absolutely. You're looking for low free energy of the interacting proteins. You want to be evaluating the energy. And moving the side chains makes a lot of sense. Fixing the backbone it's an okay first approximation. Yeah. Other things?
We talked about making random moves with the proteins and calculating the energy change after each random move, setting and rejecting it, and then running a bunch of trials.
What kind of moves would you do?
You could do changing rotamers, moving the actual backbones.
You could move the backbone.
Moving the backbones, see where they fit to each other, and getting different decoy.
Okay. Other things? Sorry. Good. Sarah and Jelyaska?
Well, it's not something we talked about, but I think shape complementarity would be the way to go if it's a lock and key model.
Yeah. So so these are all good things. Basically, the search space is basically rotations and translations of the two proteins. Which is basically 6 degrees of freedom, 3 translations and 3 rotations, plus all the degrees of freedom internal to the protein. And then you're basically looking for the low energy conformation. Grid-based shape matching algorithms. Docking has been done back since the 70s. In the 70s, Shoshana Wodak and Joel Janna did the first kind of coarse grain docking. So really, as soon as they were developing molecular dynamics, they were doing some of this, and they had to do coarse grain models, and they couldn't search very far, and energies were lousy. And then people dropped it for a while. And then this grid-based algorithms took off in 1992 by Kachalski, Katzir, and Ilya Vaxer and others. Kachalski changed his name to Katzir when he moved to Israel. He actually served as the president of Israel in the 70s. He was a scientist. Israel has this habit of using, like Einstein was the first president of Israel, so they use famous scientists often as their president. So he served as president, then went back to science and ended up doing protein-protein docking after that. He came up with this algorithm, which is kind of fun. So the basic idea is you want to match the shapes together, and they do that. A simple way to do that is to put the shape on a grid. So you can take the protein and take a shape, and then you can put that in a three-dimensional space and make a grid. And in each grid you can look at, is there protein there or not? And so you could do that for protein A. And then you can take protein B and do the same thing, put it on a grid and look at and represent the protein that way. And then what you do on this grid is each space in the grid where there's no protein, for protein A, you put a 0. So you have, let's call it, I think Kachalski called it rho. He said rho is 0 outside the protein. Inside the protein, he said rho, make that minus 15. And then for a layer about a grid length thick, we put rho equals 1. And then if the other protein, you put, oh, this one, delta equals 1 inside the protein. And then the boundary, again put delta equals 1. And then outside put delta equals 0. It's protein B. And what you can do then is take these two matrices. Essentially you have a matrix, a 3x3 matrix, a three-dimensional matrix of numbers. And what you can do is superpose those numbers on top of each other and basically take a dot product to look at how these match up. Okay, so if you take matrix A, which has 3 indices, I, J, K in space, and then you multiply that by B, I, J, K, and then if you translate this left, right, up, and down, you basically be adding to these indices. So you go I plus alpha, J plus beta, K plus gamma. So we're taking these two grids, we're shifting them up. Well, you're taking one grid and shifting it left, right, up, and down in a periodic sense. So if things drop off the right, they come back on the left. And you're scanning this grid over the top of the other grid, and you're multiplying them together. Does that make sense? So if you see what's going to happen, where there's zeros, you're going to get nothing. This is basically a 1 where there's protein. If you can overlap with the shell, if you can put that other protein here, you would have overlap with the shell, and you'd get 1 times 1 is 1 for every grid point that overlaps. That'd be good. But then if you overlapped in the core, you'd have 1 times minus 15. That'd be bad. You'd have a clash over the top. You'd get a big negative number. And basically, you're going to try to maximize the overlap in this multiplication problem. Does that make sense? So we're going to have some dot product C. That's going to be a function of alpha, beta, and gamma. That's just going to be the matrix A dotted with the matrix B. And then we want to maximize C with respect to alpha, beta, and gamma. And that'll tell you the best way you could put these together, the best translation to put these together to have this kind of glancing contact between the shapes. Because this is totally different than what we've done before, right? There's no atoms here, there's no backbone or side chain. You're just taking the shape, looking at if the one shape matches the other shape. Now, it turns out that's pretty pretty good because if you look at protein complexes, they have a really nice shape complementarity. So if you're looking at the angstrom scale of things locking together, this can work really well. The other reason it works really well is this map is really simple, right? We don't have to put the backbone and do the bonds and get hydrogen bonds. None of that matters. We're just looking at whether the shape matters. It's also really fast. Let me briefly say why it's fast. Actually, before I do that, this is going to cover the 3 translations. You also then have to rotate. So rotate, it's going to be slower. You're going to have to take molecule B, rotate it 15 degrees, remake your grid, and then do the calculation again. But you could do this for each discrete rotation of the second protein. And then you would have scanned all the rotations and translations.
For your protein shape, you also have to rotate your protein to get a different shape, right?
Yeah, so this shape, and what we would do is redo the B matrix. And we take that protein and we rotate it inside the box, but we rotate it and recreate the B matrix, and then redo your calculation again for all the translations of that orientation.
So what about a 3D dimension? You sort of...
Yeah, right, okay, so you can do this in the 3rd dimension also. You're going to translate in 3 dimensions, and then, right, the rotations, you're going to have to rotate all dimensions in 3D, right? So what you need to do is kind of take a globe and latitudes and longitudes, and you want to pick different points along the Earth and rotate it different directions, different orientations there. And you need to do corrections because all these longitude lines come together at the top, so you need to do sine cosine corrections or somehow discretize the surface of the sphere to get all different rotations in 3D. Yeah, so a lot of people are using this. There's a whole bunch of, I think Katsir's called FT Dock. One of the most popular ones today it's called Z Dock. There's also Plus Pro. One of the popular servers that uses this approach. And a lot of methods use this as the first step. What's great about this, you can do the whole protein, all the rotations, you can do a global search here very fast, whereas a Rosetta type approach would be a lot smaller. Okay, so the other reason people like this is the math can be made much faster. Has anybody done enough math? So this is basically a correlation function. You're looking at how these correlate. And do you know a fast way to calculate the best correlation? You have to have taken a little bit of math. These people heard about Fourier transform. So it turns out in Fourier space you can do this correlation really fast. So let me go through that a little bit. So in Fourier space, you can take a grid like this and you can convert it to Fourier space. If you took either A or B that has 3 indices, X, O, P, Q, is the Fourier transform. You can do this discretely by doing a sum over ln(n) e to the minus 2 pi I over n OL plus PM plus QN. So we take, times little x ln n. So this X is our A or B matrix, and it's got 3 indices. We'll sum over all 3 of those, and at each point we'll take exponential minus 2 pi I over n, and then the sum of OPQ. And OPQ are like wavelengths. If you've done Fourier transform, you're often putting things into a frequency space. So these are like frequencies, and you're doing the sum over this, this kernel here. And you end up with, instead of the small matrix A, a little matrix X, you end up with this capital matrix X that again is a 3-dimensional set of numbers. So now we end up with capital A with indices OPQ. We end up with a capital B, OPQ, and if we take the complex conjugate of that, A star indicates conjugate. Multiply these together, you get COPQ. So that is the multiplication equation. When you transform it in Fourier space, you get the multiplication of complex conjugates. And then all you have to do, so you take the two matrices, you convert them to Fourier space, you multiply them to get C, and then you undo the Fourier transform. C alpha, beta, gamma equals 1 over n squared, sum over OPQ. So this is the inverse Fourier transform. E to the 2 pi I over n, alpha plus P beta plus Q gamma. All that times capital C OPQ. So we do the inverse Fourier transform, and then we look for a sharp peak in little c alpha beta gamma. And that'll give us the best way to align these grids. Okay, so what happens in Fourier space, because you're summing over all of these possible translations and rotations, is it will do the optimization for you and find the best alpha beta gamma to make the best shape complementarity. So it turns out when you do this, well, you have to do it for each rotation, so we also include the rotations. With the rotations, this FFT is a n cubed log n cubed calculation, whereas the brute search is n to the 6th, where n is the grid size, how finely you're going to divide this up. If you have n steps of translation and rotation, you have n to the 6th search space, whereas if you do it in Fourier space, it's much quicker. It requires a lot less calculation. So this calculation is really quick. There's a lot of web servers. If you go to ZDoc or ClusPro, you can find a web server. You can upload 2 structures, and within minutes you can get back top docking decoys. So that's very fast compared to what we do in Rosetta approaches. All right, any questions about the grid matching and the Fourier space? So one of the themes here is this is kind of like, we talked about multi-scale, the centroid scale, and the full atom scale. This is kind of one scale out again, what we're just looking at a big picture of the protein. What are some disadvantages are you have this shape. It's just a simple shape. There's no chemistry here. People have played with this scheme where you just put numbers here. It just says it's occupying space. Some people have played with, instead of having this be a real number, have this be a complex number, and then in the complex space you could put the charge and have it be more positive or negative, and then try and get those to be complementary. So that's made this a little bit faster. The other thing that's tough about this is the shapes are absolutely fixed. So if the proteins come together and the side chains move, if you do this with a fine grid and you take a bound complex, you can pull it apart and you can put it back together trivially. It's really easy, and you get the right answer all the time. If you start with the unbound structures, which is what typically you want to do in a blind case and try to put them together, the side chains at least have to move. And now you're going to have things that don't exactly fit together, and then this calculation gets more inaccurate. So one thing that needs to be done is put some flexibility in here. So one thing you can do is you can make this grid more coarse, or you can lower the penalty for overlapping, so you can allow a little bit of overlapping and you can allow it to match less precisely, but then it's going to have less precision, so you won't necessarily get the right answers often. Okay, so based on all of that, and based on the fact that proteins aren't static and they do move, that's kind of what motivated us to go to Rosetta and put together a structurally based docking algorithm where we have the backbone, we have the side chains, and we can move the protein as we dock it. Turns out that's not so easy, and it's still a problem that my lab has been working on. The side chains were, well, not easy, but it was straightforward. That's what we originally did, was put in side chain flexibility, and then the backbone flexibility is one that's still a little bit elusive. We have a lot of strategies that work a little bit, but not a lot. First, we do a multi-stage approach. So Rosetta Dock. This is the docking algorithm in Rosetta. We start with two proteins, unbound structure. And then, like you mentioned, I think we can basically going to search the degrees of freedom, the translations and rotations. One of the things you can do in Rosetta that's nice is the centroid and the full atom give you two different scales. So just like in the folding, we did the centroid and the fragments and got a rough start, and then put on the full atom. So docking, we do that. We do a low resolution search. And then a high resolution search. And then this gets repeated. Let's see, I guess you start here with random start position. The low resolution search is Monte Carlo search with translations and rotations. So it's basically a Monte Carlo trajectory. You start with two positions of the proteins, and then you make small translations and small rotations. And then we know Monte Carlo, right? You do a move, evaluate the energy. If the energy goes down, we keep it. If the energy goes up, we do the Boltzmann thing. And then we repeat, and so you wander around. The high resolution, this one is Monte Carlo plus minimization. And we've talked about this with Phil Bradley's protein refinement, except now instead of moving the backbone, we're moving the protein. So the Monte Carlo minimization is rigid body moves. Which is translation and rotation. Then there's side chain packing. This is with the rotors. And then after that, there's minimization over the translation and the rotation. And then that gets repeated, I don't know, 50 times. So you know all of these steps, right? Side chain packing, minimization. Now the minimization, before we were minimizing over phi psi, now we minimize over the translation and rotation. And we used to be doing small and shear moves on phi psis, now we're doing translation and rotation. But the idea is the same. There'll be an energy landscape. We'll be somewhere, we'll move somewhere on the landscape. We'll pack the side chains. And then we'll change the landscape a little bit, and then we'll minimize to end up somewhere else, and then we'll compare the delta energy versus KT. Monte Carlo step is in here. After we minimize, we either keep or reject the move. Okay, so a low resolution search, a bunch of steps there, maybe 500 translation, rotation steps there, and then maybe 50 steps. These take a little while because they're side chain packing and minimization. Then at the end, come up with a decoy, which we talked about. So a decoy is a candidate model structure. this gets repeated maybe 1,000 times. If we know roughly where the interaction patch is supposed to be, because maybe we know some experimental information, or maybe 10 to the 6th times if we don't know anything at all and we're searching the surfaces of 2 full-size proteins. So we get 10 to the 6 decoys. Then we take the low scores, and then we cluster results. So we might get 100 or 1,000 low-scoring structures, and we cluster together which ones are similar to each other. So just by looking at the RMSD distance between each one, if we find a bunch of docking solutions around the same place, all at low energies, we like those better. So the clustering is a key step to look for convergence in the algorithm. And then from there we get to final predictions. Let me show a few slides before I do too much of flexible backbone. Sorry about the buzz, I'm not sure what that's about. So you guys know some of this because you've seen this, but here's the centroid bone. We take the backbone atoms and then the side chains, we replace them with a single point that represents the whole side chain. And then that's, you guys can see this when you do it on pi Rosetta because you can output to pi mole as you go. But these are the rigid body motions, a few angstroms at a time. It's bouncing around almost like 2 proteins might do when they're fusing next to each other. So a few angstroms, a few degrees at a time, and it's moving. Yeah.
The properties of that might differ for every interaction.
Yeah. The centroid would be different.
So like
right.
these are kind of charged.
Size on the bump score, the VDW score has a different size for those, and then the environment and the pair scores are based on these coordinates and how buried they are and what they're near. So the pairs, hydrophobics will have good environment scores when they have lots of neighbors and are buried, and then the opposite charges will have good pair scores when they're near each other. Okay, so there's the low res. These are some examples of energy funnels. Just like in folding, we can plot distance from the correct bound structure versus an energy. And what you want to see is different, each dot is a configuration, so each configuration has an energy. And in a good form of search, you have a lot of points that are close to 0, close to the native complex. And if the score function is good, then the lowest energy points will be the ones that are near native. So this is a case where this case is one where we're docking bound backbones. If we take a complex and pull it apart, mix up the side chains, then bring the bound part back together, it's fairly typical to get a nice, strong, deep energy funnel with bound backbones. If you use the unbound individually crystallized backbones, sometimes you get a funnel, as in this case. Other times you don't. These cases actually all look pretty good. Other times you don't, and there'll be changes in the backbone that prevent that from happening. And then these are some examples from Capri challenges. This is from a long time ago, but this is a blind docking of 2 proteins relative to each other, and they get, and the complexes are done by experimentalists before they publish it. They email out, in fact we're doing around right now, they email out 2 proteins and say, figure out what the coordinates are. We get 10 guesses. This is our model number 1. This is pretty good back then. We actually were given the bound coordinates of this antibody. This is a camelid antibody, and then that's an enzyme. And then other times it's model number 2 or even model number 8. This is one where we had to first fold a protein and then dock the model to the other partner. So you can see the model we got really lousy, but we have some helices in the right place enough that we can detect some of the interactions with the other protein. So that's model 6. So we had 5 models we thought were better, that had better energies, and then but were incorrect, and then the 6th model was good. Yeah, go ahead.
I actually have a couple. So, I mean, sampling, keeps being like the important or the limiting factor for a lot of this. So, what is your initial starting point for a docking optimization, especially because like I like if you start with two proteins independent of each other, and when they're next to each other, they're gonna have that proximity would have a big impact on like the changing the structure, right?
Yeah. So and in the pure blind problem, you'd get two proteins, you wouldn't know anything about where they bind. So you need to rotate this one completely, rotate this one completely, and what we do is a multi-star trajectory. Each trajectory would start from a different rotation on two spheres and then pull them together. So you'd want to be sampling every possible orientation of those two solids. That's a complete blind one. That's actually really hard. FFT is probably your best method for doing that, and Rosetta can take a long time, and the coarse grain can have some trouble identifying the best patches. In reality, in Capri, so the target we're doing now is a histone with a ubiquitin modifying enzyme, and so that it's known by reading the literature. By the time people get to crystallizing something, they've usually done some studies about the proteins, so they know because there's ubiquitination going on, there must be a lysine somewhere near the interface, and they think they know which lysine that is. So we often have enough experimental information to kind of localize things, and so then you can do what we call a local perturbation study and look within 5, 10, 15 angstroms. When we're calibrating the high resolution stuff, we cheat, and we do that on purpose. We start with the bound complex, take the unbound pieces, superpose them roughly in the right place, and then randomize by 5, 10 angstroms and rotations, and then try to recover that local funnel. That just saves computer time. There's no sense searching over here and completely missing the near natives.
Right.
So we get over the sampling problem, make sure we can prove the score function and see if the score function is working okay. Yeah, that's a good question. Okay, so in 2000, Rosetta was written in Fortran, and I didn't want to write in Fortran 77 in 2000, and so we started writing docking in a different program, and then we realized in 2000 the holy grail of docking is that if there's conformational change, if the protein fold moves, if it's flexible, people can't get the right answer. That was well understood in the literature 14 years ago. And so I said, well, we're going to have to fold the protein and move the protein. So I switched to Rosetta in Fortran and wrote Fortran for the rest of my postdoc to be able to move the protein. And then it turned out the docking algorithm I wrote didn't move the backbone because that was too hard still. But Rosetta had a much better high-resolution approach and a way of moving the rotamers that wasn't really done very much in the field, and minimizations and the combination of the two length scales. And then calibrating over large numbers of proteins to get the energy function, and also calibrating the energy function on folding and design. So all those things came together in the original docking algorithm for Rosetta, but still no flexible backbone. So my first rotation student, when I got here, I put in a problem from Capri where a moving backbone foiled us, and had that student do the flexible backbone. And that ended up being a master's thesis a few years later, and well, we're still working on flexible backbone. We tried some random things like just throw in fragments and just throw in small and shear moves, and that tended to be really difficult and really hard. So we've done a little bit of thinking in the last decade. So I want to start with how do proteins really bind? Well, really bind. Models of how proteins bind, or theoretical models of how proteins bind. So models of protein binding. So the first model goes way back to Neil Fisher in 1894, before we even really knew what all of these proteins were made of. And the model is that of lock and key. How do proteins bind? They're simply lock and key. That you have an enzyme and its inhibitor, and they have complementary shapes, and put those together, and they dock. And the shapes just match up like a key and a lock, they just match. This is kind of what we do with the rigid backbone assumption. We assume the backbones are the same, and things come together, and they don't move, and they should fit. Except for the side chains. The side chains are moving, but at the scale of the backbone, we're assuming a rigid lock and key. The second model, it's actually the third, but historically, is Monod's model, which is called conformer selection. And the idea is that a protein is not just a rigid block, but we know now that it's a fluid polymer that's in water and it's moving, it's flexible, it's moving all the time. So this model recognizes that the protein might have different shapes and it might be in equilibrium with different conformations. And all of those might exist in solution. The protein is constantly fluctuating between a bunch of different configurations. So this is a good statistical mechanics view. The other protein is the same. They also have several different configurations. And it might be that only some of these actually fit together, and the proteins might bump into each other and might not even bind because they're not in the right configuration. And what has to happen is you have to select out the right conformation from the ensemble of unbound configurations. Okay, so then this would still. All right, so that's Monod's model of conformer selection. This is 1965. Our third model comes from Koshland just a few years earlier in 1958, and it's called induced fit. And Koshland's model says that, like the lock and key, proteins are preset to be able to fit together, but then they come together into some sort of encounter complex. And that might not be, that might not be the final configuration, but after they come in contact, the interactions between the two of them cause conformational change, and then they take on a different bound configuration. Okay, so they come together in they're in one conformation in solution, and they come together, and the context between them causes them to shift to a different conformation that is the bound state. So this would be this would be consistent with crystal structures where we have unbound crystal structures and bound crystal structures, and the backbones are different. Maybe a little bit, maybe a lot. And the last model is a combined model. It's been done by a few people, which would say you would start with an ensemble of different configurations. And the second protein would have an ensemble of different configurations. Those would, conformer selection would happen first, and we'd select out the conformers that are competent to bind together, giving again an encounter complex. And then so this would be conformer selection. And then we'd have a second stage of induced fit. Here we get to our final bound complex. Questions about that? So there's experimental ways to probe this in solution. We can look at NMR data, and we can look at whether the protein is fluctuating or not, and that's been shown to be true. Proteins move. It's really a matter of extent. Some are more rigid, some are more flexible. Some have more flexible loops, some are more, some have whole domains that open and close. And then this part's a little bit harder to get. You want to look at the fast time dynamics when things come together to where they finally bind. That's a little harder to probe experimentally. But we certainly can look at the final structures and the initial structures when crystallizing alone or in complex. Yes.
especially with the conformer selection, has anyone thought about looking at the energy of the individual states? And then from the energies, calculating the probabilities of occupying each state and then multiplying those out to get the probability of the complex? And then looking at kinetics?
Okay, so you absolutely could put, you could do your statistical mechanics exercise on here. You could put energies on each of the configurations. You could make a partition function. You could say the probability of observing each configuration would be proportional to that energy over KT, or opposite that energy over KT. So you could get fractional probabilities for those. And then, yeah, you could think about from there how to go on down the line. Yeah, so the next step actually here would be, given these models, can you think about what you might do in the docking calculation to capture conformational change upon binding? So we're going a little off track from Anfinsen, right? Because Anfinsen said this is a thermodynamic problem. All that matters is the conformation at the end. And at the end, we're looking for some low energy conformation. And in docking, I guess, in flexible docking, we're going to do that. We're going to change the rotation and the translation. But now we're going to change all of the phi's, the set of all the phi's, and all the psi's, and the omegas, as well as the chi's. So we've got a lot of variables that can move. We're really optimizing the energy. We're minimizing the energy as a function of all of these variables. So we could just start randomly picking phi's and psi's and omegas. But what these tell us, these are kinetic pathways. So this is, Anfinsen is thermodynamics. It's really about the final energy and the beginning energy. These are kinetic theories that tell us about the pathway to the final structure. So why is that helpful? In docking in the computer, we need to come up with a pathway to go and calculate the final configuration. So how do we go about doing that? So lock and key, I'm going to do translations and rotations and look for the lowest energy configuration. How can I do conformer selection in the computer?
So you generate a bunch of different protein combinations with different side chain position and background position?
Exactly. So I could start with one configuration, and then I can generate a bunch more configurations from there. So the first thing I need to do is generate a bunch of conformations with different backbones. Okay. So the way we do that is just by running the relaxation stage of Rosetta, the high resolution refinement that you've already done. Small and shear moves, packing and minimization. You can start with the crystal structure and just shake it around a little bit. And so we do that, we'll generate 50 models and then take 10 low energy models that might be more represented in the partition function. And then what? Then we have to dock them. And you could, one way to do it would be simply dock all of the combinations, which could get expensive because then you could have n models here times n models there. You have n squared docking calculations. That's one way to do it. What we actually do is while we're docking, we're doing rigid body translation rotation moves. Then we also do backbone swap moves where we take out backbone 1, we put in backbone 2. We take out backbone 2, put in backbone 3. So we're doing rigid body moves, and then we're also swapping out from pre-configured backbone configurations on both sides. So it's essentially, we've taken all of these variables, the backbone variables, and made a library. So like the Rotamer library, like the fragment library, we made a backbone library for each protein. And we do Monte Carlo backbone swap moves. So it's a real simple way to extend the Monte Carlo algorithm. One of the strengths of Monte Carlo is we can just put in different random moves, and now this move lets us move around backbone space. So just like with the Rotamer library, there's some limitations. If you don't have good Rotamers, you might not find the right thing. If we don't have good backbones, we might not find the right thing. But it turns out in the data we do, for small backbone change, if there's less than an angstrom backbone change in the proteins, then having an ensemble and docking an ensemble helps us get the right result more often. I might have that data on the slide. Okay, so then the second model, induced fit. How would we do this? That again tells us the kinetics and the steps that we would do in a computer algorithm. So this looks the same. We dock stuff together. And now we need to look at how the energetics with things responding to each other move things. So what we actually do is take these encounter complexes, and in the minimize step, when we minimize, usually we're minimizing over R and T. We also minimize over the phi and psi angles. So in the minimize step, we let the backbone move according to the gradient in energy and just go downhill. So we're not really searching every backbone configuration, we're just going downhill in energy. And well, in order to do that, you first have to start with the unbounds and go downhill without the comp partner there. So this actually might not be the crystal structure, it'd be some kind of minimized crystal structure. But then here, when you minimize, because of the presence of the atoms on one side, it's going to affect how things move on the other side, and they'll move in response to each other. Make sense? So we actually do the conformer selection in the low res, because we're doing wholesale swaps of the whole conformation out. And then this one, because we're doing the minimization and we're looking at the finer details, we do that in the high res. So then it's really easy to do the combined model. We just turn on the ensemble docking in the low res and turn on the minimization in the high res and put those two pieces together. So let's see. This is how we generate an ensemble. We pull off the side chains, idealize the bond lengths and angles, because we know the refinement. We use idealized bond lengths. We make 10 low resolution structures, and then in high res, and then refinement. So we get about that much variability. So we tested this on small conformational change, like under an angstrom. Over an angstrom, still involved. It's hard to do this. You can see in the relaxed, you're still getting just small changes in the conformation. Okay, then in the conformer selection, we put all of the proteins on top of the current configuration, make a quick partition function, and pick a particular backbone to move ahead in the search. And here's some detailed results. Basically what happens is we see better funnels, we get better sampling when we do the conformer selection, because we're getting a broader range of backbones. And with the minimization, what happens is the energy function funnels get deeper, so we get better lower energies. We also get lower energies for the false positives. Sometimes the bad decoys will get low energy also, but when it's working well, you get nice deeper funnels. This is the fraction of native contacts correct in that one also can go up dramatically, these minimizations. This is a test set of 21 crystal targets. If you take the bound, pull them apart, put them back together, most of these will dock correctly. If you do the unbound and use fixed backbones, you lose about half of those cases. Conformer selection gives you some of them back, and induced fit gives you some of them back, and the two of them together give you more. The other fun thing we can do is I don't know if we've looked at yet, an NMR structure. If you download an NMR structure, they're based on shifts in the NMR peaks. Typically, those shifts don't give you a unique model, they give you several models for the structure. So if you download an NMR structure, you'll actually see 50 backbones and some variation between them. It's easy to look at it and go, well, that's a flexible protein. It's not, it could be if the protein's flexible, but what it really means is the experiment didn't give you enough data to lock it down. So usually the terminus is flopping around because there's not many couplings to the terminus, and so you don't lock its position down. So you see all that indeterminate in the structure. What we can do with the ensemble conformer selection approach is you can just throw all those models in and dock them all at the same time. So one of the things we got for free when we did this is we can dock all of the models from an NMR structure, and we tend to get much better results here than when we just put in a single model from the NMR. Here's an NMR model we did in Capri. So here's an NMR structure. Here's 60 models, and you can see the terminus flails about, the loop flails about. You know, realistically, that might be the flexible parts of the protein, but that's not what the data mean. You can't necessarily conclude that. When we dock them together, we've got many good models, and we have one of the best models of any group in the challenge, I think, because we have this ensemble stuff working. Yeah.
Why do you run an ensemble instead of constraining the residues based on the NMR data?
Well, the ensemble is faster. I have 60 configurations rather than moving it around. I have a limited, I'm using that library, I'm assuming that library is good structures, just like the fragments we assume are good structures and the road merge we assume are good structures. But you're right, it's not fail-proof. Yeah, it's a trade-off. This is one where we docked and the motion of the loops was very critical. The minimization played a big role there, and also some loop building, which is our next topic. So we'll move that to transition. Any more questions about docking? Okay. So, I guess there's one other last method if you're looking at docking that you might see in the literature for flexibility. Flexible docking is called normal modes. And what you do in normal modes is you take the protein as a set of atoms and you put springs between the atoms. You can do this either in the complex or in the each individual protein. And if you take that set of atoms and put a bunch of springs between them, so the energy of the spring would be K. The energy would be kx squared, or where kx minus x bar squared, where there's some length between the springs and then they vibrate. Okay, so essentially a spring on its own will vibrate. If you put many things together, you can see the vibrations of a large body. If you have a, say, what you end up seeing when you look at how these springs would vibrate is you see these low frequency motions. So for example, if you had two lobes, one of the ways this would vibrate is it would open and close. That's a standard way. If you think about the way a solid would distort or vibrate, that's the same kind of math you're doing. So I don't want to go deep into the math, but what people will do is they'll calculate normal modes and say there might be a stretching motion, and maybe a twisting motion, and maybe an opening and closing motion, and they'll put those particular motions into the docking calculation. So again, instead of sampling all phi's and psi's, we're sampling along directions of the lowest frequency normal modes. This tends to work well if that is the type of motion you see. So maltose binding protein, when it binds maltose, the small molecule, it opens and closes. So this works really well for that kind of docking. It's not great for small scale motion. So if there's a loop that flops around or a terminus that flops around, or some smaller side chain motions, you don't get those in this large coarse spring model. So it's really for capturing large scale motions. Large scale motions. All right, I think that's all I'm going to say about that. So there's a few groups using normal modes for flexible docking. There's a few groups doing all-out molecular dynamics for flexible docking after they get in counter complexes. That takes a long time. It's very expensive. Has the same issues with energy function, whether it's the energy function is really going to have a minimum or not. But this is still an open question, especially if the protein has major motions, if pieces of it unfold. Then that's something that's difficult to predict, because you want to start with the protein folded, like you know in the unbound state. As soon as you start moving it, the space gets really large and you tend to wander away from it.